Hello everyone, it's Chemicator. Welcome back to my channel. Today I wanna talk about molecular editing, a modern technique shaping the future of organic synthesis and revolutionizing drug discovery. Right now, organic chemistry has reached a point where we can build almost any molecule. So the most important question is, how do we build them? Laboratories around the world are focused on finding more efficient methods for synthesizing target molecules. As a result, the toolbox of organic chemistry has expanded significantly in the past few decades. Innovative strategies such as photochemistry, electrochemistry, click chemistry, CH activation, flow chemistry, and microwave chemistry have greatly advanced the field. However, in practice, only a limited number of reactions are widely used in drug discovery and development. Molecular editing is a concept that helps organic chemists address unsolved challenges in synthetic chemistry. To better understand this approach, let's compare it to a child Tinker toy set. Now look at this tower. How will you change its structure? It's obvious that the best way would be to remove the part you don't want and replace it with a new one. But this approach doesn't work for molecular structures. Take this drug for example. It's a kinase inhibitor. Studies show that replacing this carbon atom in the ring with a nitrogen atom significantly improves its pharmacological profile. Now imagine the entire structure as a tower where we need to pull out the carbon atom and insert the nitrogen. Traditional methods would destroy the entire structure, but molecular editing allows us to make precise changes with high efficiency. And that's the power of this approach. In other words, if we wanted to synthesize a modified drug with a nitrogen atom in its structure, we'd have to build it from scratch, which is not financially viable due to the long synthesis route. Molecular editing provides a game-changing solution to this challenge. Generally speaking, molecular editing can be divided into a skeletal and peripheral editing. In a skeletal editing, the core scaffold of a compound undergoes changes, which may involve the modification of a single atom or multiple atoms. In peripheral editing, the core structure remains unchanged, while various functional groups are installed on the skeleton. Now let's start with two classic examples of molecular editing. In the Favorskii rearrangement, an alpha-chloroketone is converted into carboxylic acid derivatives. This is an example of ring contraction, where a 6-membered ring is transformed into a 5-membered ring. First, the hydroxy ion abstracts a proton from the alpha position of the carbonyl, generating an enolate intermediate. The resulting enolate then attacks the carbon bearing the chloro group, forming a cyclopropanone intermediate. This intermediate then opens up as the hydroxide ion, now acting as a nucleophile instead of a base, attacks the carbonyl group. Another classic example of ring contraction is the Benzilic acid rearrangement, in which the hydroxide ion attacks one of the carbonyl groups. In the next step, the intermediate undergoes a 1-2 shift, where a carbon-carbon bond migrates to the second carbonyl group. After protonation, the carboxylic acid product is formed. Notice that these two classic examples involve compounds with carbonyl groups under basic conditions, and in both cases the editing process results in ring contraction. However, molecular editing can also lead to ring expansion. So let's explore ring expansion with a more complex example. Substituted benzene rings are a crucial component of many approved drugs and agrochemicals, including aspirin, ibuprofen, acetaminophen, and amoxicillin. The significance of this motif makes it the most prevalent ring system in pharmaceuticals, and a highly attractive structural target in drug design and development. However, the metabolism of benzene rings under physiological conditions can generate toxic intermediates. A common strategy to address this issue is replacing the substituted benzene motif with saturated polycyclic hydrocarbon scaffolds. BCP, Bicyclo 2.2.2 octane, and Cuban derivatives are well-known bioisosteres of benzene. These structures effectively mimic the topological pattern of substituted benzenes while enhancing the metabolic stability of drug analogues. For example, in 1996, Pellicheri discovered that the BCP-containing amino acid was 3 times more active than its benzene analog as antagonist of group 1 metabotropic glutamate receptor. In another case, in 2012, Antonio Stepan replaced a fluorophenyl moiety in a γ-secretase inhibitor with a BCP unit. This bioisosteric modification improved activity, water solubility, metabolic stability, and reduced lipophilicity compared to the benzene analog. It's important to note that benzene ring substituents can be positioned in ortho, meta, or para relationships. Until recently, most research focused on mimicking para substituents benzene. However, advancements in meta and ortho bioisoesters are now emerging. In this regard, Pavel Mikhailik has identified difunctionalized Bicyclo 2.1.1 hexane as an attractive candidate for both ortho and meta substituted benzene bioisoesters, depending on its substitution pattern. His group successfully replaced a benzene ring with BHC in Valsartan, an antihypertensive drug, and Fluxapyroxad, a fungicide. Despite this progress, synthesizing dike-substituted BCH remains a significant synthetic challenge. In 2023, Professor Proctor presented a catalytic approach for creating this challenging target. In its reaction, the alkene inserts into the central bond of Bicyclo 1.1.0 butyl ketone. The starting material is readily accessible. In the first step, allyl chloride reacts with bromoform to create 1,1-dibromocyclopropane. This then reacts with methyl lithium and tert-butyl lithium. This intermediate reacts with Weinreb amide to produce the desired ketone. Adding alkene and samarium diiodide result in the target molecule. Now let's go through the mechanism. In the first step, SMI2 reduces the carbonyl group by single electron transfer, resulting in the formation of a ketyl radical. This carbon-carbon bond in the bicyclic intermediate undergoes homolytic cleavage, generating a planar cyclobutyl radical. At this stage, the radical intermediate attacks to the olefin, forming a stabilized radical intermediate, which then undergoes a radical rebound to give a new ketyl radical. In the final step, another single electron transfer generates the target molecule and regenerates SMI2. The power of this method is reflected in the broad range of diversity it offers. For example, by selecting the appropriate ketone, we can incorporate ibuprofen or naproxen skeletons into the BCH. Similarly, a wide range of derivatives can be accessed by modifying the olefin structure, such as menthol and vitamin E.